Okay, so today is our 13th lecture. So we are finally entering the module where we explore more advanced topics. So we finished the transcriptomics and we know about how we analyze gene expression data. But those are applied to the bulk of the tissue. So we extract RNAs from every cell in the sample and we analyze the gene expression together. But in many situations, we require knowledge of individual cells in the sample to understand the biology. So today we're going to focus on two techniques that will allow us to analyze omics data at higher resolution than just the bulk of the tissue. So this will be again a two lecture kind of series. So today we are going to focus on the technique, the omics techniques, and some kind of data processing. Next week we are going to talk about the detailed statistical model and the bioinformatic tools that we are going to use. Okay. Just to give some motivation, so there are many situations where if we analyze the tissues or the sample together as a bulk, we only identify the majority of the gene expression or the mutation. Because when we analyze all the cells together, what we are getting is an average. It's not individual cell, it's an average. So for example, if you study patient and you identify from RNA sequencing like certain gene expression, and you want to target those genes with drugs, your drug is going to be effective against only the average, the majority of the cells, and not every cell type in the disease tissue. What we are talking today, the first technique is called single cell technique. So SC here is single cell. So we're going to see approach where we isolate all the individual cells out from the tissues. We analyze each of them separately. So we get information about the gene expression profile or the mutation profile for individual cells. So we know like how many cells are there in the sample and what type of cells are there in the sample. So we can make much better decision, or we can learn better about the biology, because each biological system is not a single cell type. Many of them consist of multiple cell types. And by being able to deconvolute or decode the composition of the cells, that we can understand the system. So talking about the kind of disease model, if you think about cancer, like the tumor, the cancer is an environment. It doesn't consist of only cancer cell itself. It also contains neighboring cells, like maybe the blood vessels, maybe the immune cells that are recruited and change also because of the transcriptomic change in cancer that communicate to other nearby cells. So when people study cancer in the past, like when we only have bulk analysis, we analyze gene expression, mutation profile of the whole tumor, like, or maybe some part of it using the biopsy. But we don't really understand the molecular changes that occur inside the actual cancer cell. We only see the average. So for example, if you analyze any cancer sample, like transcriptomics from cancer tissues against normal tissue, the first thing you're going to find is an enrichment of immune genes. And that's because in cancer environment, you have immune cells. But in normal tissues, you have less immune cells. So you're going to see an upregulation of immune-specific markers in cancer, regardless of cancer type. And that's just because the sample contains immune cells inside more than usual. That interference can only be removed if we can study individual cells specifically. So in cancer research, when people talk about intrinsic change, that means they want to focus on the change on the actual cancer cell. So they want to remove any transcriptomic, any mutation profile in other cell type. They only want to focus on the actual cancer cell. And when people talk about tumor microenvironment, these are the other cells, non-cancer cells, even bacteria that populate nearby the tumors. And bulk omics cannot disentangle these two things. We don't know that each upregulated genes, if it's intrinsic to the cancer cell, or is it based on the change in the environment of the cancer cell? Another situation that several people are interested in, so inside each tumor, there will be some specialized cell, they call it cancer stem cell. So it behaves like a stem cell. It can differentiate, it can create a whole new cancer if the cancer stem cell is alive. But if you do a bulk omics inside tumor, there will be very few stem cells, and most of the cells will be the differentiated version. So when you do bulk omics, you only see the signal from the differentiated cancer cell. And if you decide to kill those cells using any drugs or chemotherapy or radiotherapy, if the cancer stem cell is still alive, it will regenerate. So you can have relapse. You can have a new cancer again. So people want to be able to identify, to pinpoint signal of stem cell inside the cancer tissue. And because they are a minority population, the only way to do that is to study individual single cells inside the cancer. So on top, in addition to the kind of disease or cancer system, knowledge as single cell resolution will tell us basically in any biological system that you are analyzing, you will know the heterogeneity, what cell types are in the sample and what proportion. If you want to study the evolution of the cells, like if you culture the cells for many days and you want to see if there is any mutation that would occur along the way, if you analyze the culture at single cell resolution, you can track the offspring or the daughter cells from every original cells. So you can trace the clonal expansion, you can trace the lineage. When you want to study like stem cell system, like where some cells differentiated, some cells remain stem cell, you want to understand the composition, and you want to see the transcriptomic change that occur between between stem cell and differentiated cell. In this case, you need to have transcriptomics data at individual cells level in order to compare the genes that are upregulated or downregulated during differentiation. And particularly, we will think more about this next lecture. But one big benefit from single cell analysis is that we can reconstruct cell development. So usually, if we want to study cell development, we need to culture the cells. We need to collect the sample at multiple time points, maybe at day 0, day 1, day 3, day 5, day 10, in order to know the cell development. But with single cell technique, one thing that people found is that in our tissues, not all the cells are differentiated. Sometimes you have fully differentiated cells, sometimes you have partially differentiated cells, sometimes there's still a small population of stem cells in the sample. So by isolating all the cells and collecting all the omics data, you are essentially seeing the transcriptomic program all the way from stem cell to differentiated cells. So inside your single cell data, you can imagine that you're getting transcriptomic signature from different stages of cell development that are mixed inside your tissue as individual data points. So if we are able to order the cell, maybe by similarity in their gene expression, like stem cells are similar to other stem cells, but not similar to differentiated cells. And differentiated cells may be similar to partially differentiated cells, but different from other cell types. If we can use this kind of similarity to order the cell, to sort them into something that looks like this, where we have stem cells on one side, and we have differentiated cells on the other side, and cells that are in between, the development, are sorted in between these two. If we can do this, and we will learn how to do this next lecture, then we can reconstruct the whole development pathway. We can identify genes that are upregulated in a certain lineage. So in the future, if you want to differentiate cell into a certain cell type, you know which gene to turn on, you know which gene to turn off. So this has a lot of benefits for biologists and people in biotechnology. Okay, but for this lecture, we're going to focus on how we can do single cell first. Okay, so typically, as the name suggests, if we want to analyze the data at single cell resolution, first we need to have a method to isolate all the cells. So in the past, it started with manual. You have to pipette all the cells individually, maybe through the guide of the microscope. You can see all the individual cells, and you can select them out to analyze. Then people came up with facts using fluorescent surface signal to sort the cell. Because different cell types will have a different surface markers. So we can sort them into batches, into groups, based on their surface marker signature. But that still rely on the knowledge of the cell surface marker. So modern single cell pipeline is fully automated. We don't need to know about how many cell types are there or what protein markers they have. With microfluidic device, you can flow the cells into from one direction, and you can flow your PCR and sequencing buffers from another direction. And you control the volume of the cells, the flow rate of the cells, in such a way that each doublet of sequencing and PCR reaction that forms will, on average, most of the time, it will contain only one cell. So you can isolate all the cells into different individual doublets. Okay. And once you can do that, like in each individual doublet, so this will be contain all the enzymes that we need to do the sequencing and polymerization reaction. So each cell will be if we do RNA sequencing, so the RNA in each cell will be reverse transcribed, will be PCR amplified individually. So each droplet contains RNA from one cell. And the trick is, if we have to do sequencing separately for each cell, that will be costly because you have to do sequencing many times. So the trick that they are going to do is they're going to, in that PCR adapter, they're going to incorporate some kind of barcode, similar to how you do multiplex experiment. You do barcode individual sample, you can mix them, sequence them together, and then you can decode which read comes from sample 1, which read comes from sample 2 by looking at the barcode. So in single cell technology, in their PCR handle, they are incorporating the cell barcode. So in each droplet that contains one cell, all the PCR reaction in here is going to have the same barcode. So every RNA molecules from these cells is going to be attached to the same cell barcode. So you can use this cell barcode to assign each sequencing read to individual cells. Another feature that they add with single cell sequencing is, so we learned from the transcriptomic session that when we do RNA sequencing, we PCR amplify the RNA, so we can have amplification bias. Some genes are amplified better than the other. So the read count has this bias being added. The best thing you can do to count gene expression is to count the number of molecules. And we saw this with the nanosync technology. They attach the fluorescent probe to individual molecules of RNA. So you have the molecular count, not the read count. So in single cell sequencing, in order to get the actual molecular count, not the read count, so each PCR handle, each PCR adapter, not only contain the unique cell barcode that's unique to the cells. For each adapter, they will have its own random UMI or unique molecular identifier. So for each original RNA molecules, it starts off as one molecule. When it's PCR amplified, it's going to be attached to a random UMI, which is also a nucleotide barcode. The thing is, each original RNA is going to be amplified and attached to a unique code. So if you have 5 RNA molecules for the same gene, if these are from the same gene but different RNA molecules, they will be amplified with different UMI. So in the end, when you have the read data, every read that have the same UMI comes from the same RNA molecules. So you can collapse. If you have 5 reads that have the same UMI, instead of counting the gene expression as 5 reads, it will be counted as just one UMI. So you can get the number of molecules from using this kind of nucleotide barcode. And these two techniques allow you to generate PCR amplified product that have both the cell signature and also the molecular signature. So you can combine all the amplified RNA, DNA, cDNA, and then do the sequencing together. You can do a pool sequencing. You don't need to sequence every doublet separately. You can sequence everything and then use bioinformatics software to read out the barcode, to group the cells, and to read out the UMI to get the molecular count. Okay. So one thing that will be different, a big difference between bulk transcriptomics and let's say single cell transcriptomics is the amount of data that you are getting. So usually in bulk RNA sequencing, like in bulk transcriptomics, you have replicate. You maybe culture the cell multiple times, you get the RNA from multiple patients. You get one number for each gene, for each patient, like for each sample. You measure a gene only one time. When you do single cell sequencing, so each of your culture, each of your sample is going to contain thousands or more of individual cells. So your data, you can imagine, for only one sample, you're going to have about 3,000 to 10,000 cells. In your data, this will be columns. You might have 3,000 to 10,000 columns. And in each cell, you're going to have some sequencing reads that are from those cells. So imagine like when we do transcriptomics, I did mention this explicitly, but usually we sequence in the order of 10 million reads per each sample in order to get a good quantification for individual genes. With single cells, even if you increase the number of reads, in this case like 150 million, so you're sequencing 10 times more than the usual bulk transcriptomics. But because you have 5,000 cells, when you divide the number of reads to the number of reads per cells, it's going to go down a lot. And usually you have only 20,000 or 50,000 reads per cell. So you can imagine that when in human we have thousands of genes, and we even have more isoforms of RNA. But if you do sequencing with only 20,000 to 50,000 reads, you get a good quantification of the gene expression. If you have so few reads, how can you measure the expression level in detail? And the thing is, you can. You only get an approximate expression level. But for the purpose of single cell analysis, the purpose is usually not to identify actual gene expression. The purpose is usually to understand the cell composition. You want to identify cell type, and you want to understand how many there are. And knowing and having just 20,000 to 50,000 reads is usually good enough to identify if this is an immune cell, if this is a T cell or B cell. You don't need millions of reads to say that a cell is a B cell. You can see the signature of these cells by using only this thousands of reads. So single cell is still viable, but it has a different purpose than bulk transcriptome. And of course, you are going to see less genes. Instead of seeing all 20,000 genes, you might see only 3,000 because you have only 50,000 reads. You're not going to be able to detect all the genes. So this can create a problem. Single cell RNA sequencing is going to be less sensitive than bulk sequencing. In bulk sequencing, we combine multiple cells. So even if we are analyzing genes that are highly expressed or low expressed, we are always going to detect them because we are combining RNA material from thousands of cells together. You're going to detect almost everything. But when we do single cell RNA sequencing, cells that have high expression, we might saw the gene. But in cells that have low expression of that gene, we might not see anything. So we're going to have many missing values in the data. And this is something that people about 10 years ago, when they first started to develop single cell sequencing, this created a lot of problems for bioinformatics because all the statistical models that we learned so far, none of them works well when you have many zeros. Any distribution that we learned before, like normal distribution, negative binomial distribution, Poisson distribution, those doesn't allow for a lot of zeros in your data. It expects that you have all the measurement, most of the measurement in your data. So one thing that people found is that everything we learn about differential expression doesn't fit single cell data. So people have to come up with different techniques, different ideas to tackle it. So just to summarize, so this lecture and next lecture, we are going to go through how we analyze the data. But just to summarize what we are facing when we switch from bulk technique to single cell technique is that first, we are going to have a lot of zeros in the data, a lot of missing values. We need to do some imputation, or we need to update our statistical model. The second issue is, now that we see all the individual cells, they are not going to look the same. When we think about replicate in bulk transcriptomics or bulk omics, we expect replicate to be very similar because we are averaging over thousands of cells. Of course, the average is going to look very similar when you repeat the experiment. But now that we are seeing the individual cells, even cells from the same cell type, each individual cells are not going to have exactly the same expression profile. So we are going to see biological variation in our data, not just technical variation. So we are seeing more variance in the information. And because we are sequencing only 20,000 to 50,000 reads, we're going to have chances to detect or not detect some genes. And the expression level also change from cell to cell, depending on their biological state. So what we are seeing here is that we're going to see also a very strong batch effect. If you analyze the same tissue twice into separate single cell sequencing, you can get different data. Because you have low read count, like you are basically sampling the gene with very few read count, so your data is going to be very noisy. So this is a big problem that you need to do. If you have batch effect, you cannot interpret anything because everything will be confounded by the batch. And the one that I highlight, this is something that we couldn't do with bulk transcriptomics, but we can do with single cell. And that is the state of the cells that we are studying. In bulk transcriptomics, we are used to having treatment versus control, like very discrete condition, or maybe cells from day 1, day 2, day 3, very discrete condition. But when we are analyzing single cell data, each cell is going to have a different state, different continuous state. Let's say from stem cell, like being non-differentiated, from partially differentiated to fully differentiated. Cells are not discrete. The gene expression inside the cells, as they differentiated or as the disease becomes more severe, the gene expression is going to change gradually. It's not like an on-off switch. So now, when we compare gene expression data from cell to cells, it's not going to be clear-cut. It's not going to be group A versus group B, but it's going to be from 0, 0.1, 0.2, 0.3, all the way to 100. The state is going to be continuous. And we'll see this in the next lecture, how this is, it looks like a challenge, but it became a benefit for biologists. The last one, in terms of computation, the data is going to be much larger. So imagine if you do a transcriptomics with 10 samples, you identify 10,000 genes each, so you have about 0.1 million measurements in your data. Let's say you perform a single cell analysis. For one experiment, for one sample, you might have 5,000 cells. And you identify 3,000 genes. Okay. This means you have 50 million measurements in your data. And with bulk analysis, these numbers come from having 10 samples. So it's your whole experiment, equal your whole study. But for single cell, this example is just one sample, like one patient. You already have 15 million numbers. So your data is going to grow a lot, and you need much more powerful computers to analyze the data, and you need a much better algorithms to analyze the data. Otherwise, it's going to take days to process the data. Okay, so today we're going to see some quality check and processing techniques to appreciate the change in data type and data size from bulk to single cell. Okay. So in single cells, when we process the data, and you will see that this will be totally different from bulk data. So first, because we isolate like individual cells into droplets, into individual portions, sometimes those droplets can contain more than one cell. And sometimes those droplets contain no cell at all. So we need to remove these noisy noise data from the result. So we can imagine that if we have no cells, like if there is a droplet and there is no cell in it, you're not gonna get many read counts with that cell barcode, because inside that droplet, inside that cell barcode, there is no RNA molecules. You're going to get low read count and low gene count when you have no cells. And if you have more than one cell inside the droplet, because you have more RNA material, you're going to detect more genes. You're going to get more read count. So you can use these basic information to try to discriminate what are my actual data from cells, what are my noise. Another issue is when we analyze all the cells, that also includes dead cells. And dead cells are problematic because when the cells die, like their transcriptomics profile will stop functioning. So you get a kind of weird gene expression profile. So we want to remove this. One of the signatures that people use to remove dead cells is high mitochondrial expression. Because when the cells die, the other genes stop producing, mitochondria can still have high expression. So if we see a high proportion of mitochondria genes, that can give us some signal that these cells may be dead. But as I put in the parenthesis here, some spatial cell type, as I will show with actual data, some spatial cell type also have high mitochondrial expression. So when you remove cells based on mitochondrial expression, you have to make sure to check other cell type markers as well, to be careful about this. The next step would be how to normalize the data, because we have very few read count, low read count, so the data is going to be very noisy. How can we normalize it? How can we standardize it? And the last one, for the processing stage, will be how to correct for the batch effect. Okay, so the first technique. So the technique that I picked here, I picked because it will allow you to see certain things that you can generalize to other situations. So the way that people use to identify empty droplet, so the droplet with no cells. So the assumption here is that empty droplet should have very low molecular count because it doesn't contain RNA. So if you sort your data, you group all the reads into cells based on the barcode. So the x-axis here is the barcode. You sort it from barcode with high molecular count, so these could be real cells. And you sort them by molecular count, so this will be lower and lower until at a certain point. What we are observing here is when the molecular count goes from 10,000 all the way down to 50 really quickly. So these are barcode or droplet that only have 50 or less kind of molecule of RNA. So people identify this shoulder, it looks like a shoulder. So this shoulder where you have a sharp drop in molecular count from more than 10,000 all the way down to 50. This sharp drop kind of gives us the signal that maybe these are cells and these are not, because you have a sharp difference in the number of RNA molecules. This is related to another technique that you might have seen somewhere else called the elbow method. It looks like an elbow. So this looks like a shoulder, but if you flip it upside down, it also looks like an elbow. So people sometimes call this elbow method. And the idea is when you plot a graph of a certain behavior or certain properties of your data, if you identify a certain point where you see a change in slope, from high slope, very steep, to very flat. Usually this tells you that maybe this is where the data and noise are separated. Or in the future, we will see that this method can be used to identify model parameters. If you adjust the parameter of the model and you measure some result, and you see a sharp drop, and then change it to a flat area, this might be the appropriate parameter. So this is also similar if you recall the metagenomics section. When we want to understand whether we have enough sequencing data for a metagenomic sample, we plot the number of OTU as together with the read count. And we want to identify samples where the OTU converge, start to plateau. This means that getting more read doesn't detect more OTU. Versus other samples, if you have a very sharp increase, which means that if you get more read, you will see more OTU. That can help you identify good sample from bad sample. So in this case, by plotting the number of molecules count and the number of RNA versus cells barcode, we sort the cells based on molecule count, we can identify where real data and noise change. So we can identify that every cell above this point, we can probably analyze. Every droplet below this point, we can discard from the data. Okay. Another way that we can quality check the single cell result is to visualize the mitochondrial expression, gene count, and read count together as a scatter plot. So on the left side here, we plot the gene count. I think I start to forgot kind of N count and feature. One of them is a gene count, one of them is a molecule count. So if you plot the let's say gene count versus the percent of mitochondria, and you see this pattern. These cells that are above this line, their property is that they have high mitochondrial expression, 10 to 20% mitochondrial expression, but somehow you don't detect other genes. The gene count is very low, and most of the expression are mitochondria. So this is a clear evidence that these are dead cells. In dead cells, you don't see other genes, you see mostly mitochondria. So you can apply a filter, maybe a line at 5%, and say that anything above the line, these are dead cells. For the other example on the right, so we plot the number of molecule counts against the number of genes. And we see some outliers here that have almost twice or 3 times the number of genes and the number of molecules. So these might be droplets that have more than one cells. So you see more genes and more RNA. So you can maybe put a threshold here at 2000 genes and say that anything above this might be droplet or droplet with multiple cells. And one thing that you will notice here is that where to put the threshold seems very subjective. There's no statistics involved. Right? People just look at this by eye and then put the cutoff. There are tools that try to apply statistical model to make the cutoff more objective. But because people allow you to do this by eye, because you are going to have 5,000 cells, and you are removing maybe 1% or 5% of the cells, it will not matter much because most of the data is still going to be driving your analysis. So whether you push the threshold up and down, like 5% to 6% or 5% down to 4%, it's not going to change your result much. So that's why people are not picky about where to put the threshold. But you should visualize the data and see if you have to put the threshold somewhere. And this is the example that I mentioned that you can have high mitochondrial expression. So in this case, the mitochondrial expression is on the x-axis. You can have high mitochondrial expression and also high gene count, high molecule count. So this means that it might not be dead cells, because in dead cells you expect high mitochondria and you don't see other genes. But in this case, we see high mitochondria together with also high molecular count. So it could be some spatial cell type. And in this case, we try to apply some stem cell marker, and we found that these cells are indeed some spatial stem cells that have high mitochondrial expression. And if you do some literature review, people will say that they also found high mitochondrial expression in cardiac cells, in muscle cells as well. So basically cells that need to produce energy, you can see high mitochondrial gene expression. So before you remove high mitochondrial gene from your single cell data, just make sure that they might not be a spatial type of cell that you might be interested in. Okay. So after we filter out dead cells and other droplets that we don't want to analyze, the next step would be how to deal with the missing data, how to deal with the noisy data, the expression level that we get, how can we stabilize them. So people propose several ideas. So 10 years ago with this, people proposed several ideas to handle this. Some people say that, can we do an intermediate? So the bulk transcriptomic, we clump all the cells together and we get one measurement. For single cell, we isolate all the cells and we get all separate measurements. Some people say that maybe we have too many cells and we have a lot of signals. Why don't we combine some of them? So this will be like a local average. You combine 3 cells together and compute the average. You combine 4 cells together and compute the average. This will help because when you combine multiple cells, you combine cell with no detection to cell with measurement. When you average, it's going to remove the zero. You get some numbers instead. Other people say that, why don't we use the information that genes with similar expression level have similar noises? We saw this before in the DEC slides, where we saw that when DEC2 model want to estimate the variance for each gene, they estimate the variance for genes with similar expression together, not individual. So if gene A, B, and C have low expression level, the tools can use information from all these 3 genes to correct each other. If gene D, E, F, G have medium expression level, we can use data from these 4 genes to correct or to normalize the gene expression together. And to show you some visual evidence, so in bulk RNA sequencing, so people try to normalize the gene expression. And in this plot, they are showing the y-axis being the normalization factor and the x-axis being the expression level of the gene. So in bulk RNA sequencing data, the normalization factor is stable. When you have more sequencing depth, like more reads, you still have the same scaling factor, the same normalization factor. And this is because you average the data across multiple cells. Your measurement is going to be very stable. But in single cell data, if your cell has low expression, medium expression, or high expression, that the scaling factor can change depending on that. So grouping the genes by their expression level can give a better correction factor than doing them individually or doing them together with a single factor. So these are what people found when they try to make sense of the noises and variance in single cell data. Another idea that sounds interesting is, why don't we just model the process that we get the missing value directly? So when we have missing value, it comes from 2 factors. What's the actual abundance for the gene? This is the expression level of the gene inside the sample. And depending on that expression level, our sequencing process have a chance of detecting that gene. For example, if the genes are highly abundant genes, we are going to detect them most of the time because we are going to sequence them consistently. But as we are looking at lower and lower expressed genes, the chance that we are going to detect them with just 20,000 to 50,000 reads is going to go lower and lower until for low abundant genes, you might not see them at all because you don't have enough sequencing depth to detect this gene. So people model the abundance that we are seeing, like the expression data that we are getting, is a function based on true abundance, like how high the expression actually is, multiplied by the probability that we are going to detect it. If we detect it, then we see the true abundance. If we don't detect it, then we got zero. So we can separate this observation into two processes. So people also fit this kind of model onto single cell sequencing data. So after we can normalize the data and we get rid of all the zeros, now we want to remove the batch effect. We want to be able to analyze data from different sequencing runs, data from different patients, data from different conditions together without being confounded by the batch effect. And this example will show you clearly, even if you analyze the same data twice with single cell techniques, you are not going to get the same result. So these are two analyses of the same data. When you visualize it, we're going to talk about the technique that we can visualize this data next lecture. But when you visualize it without doing any batch collection, the data will look like completely different sample, even though these are from the same sample, just analyzed twice. What we want to see is this. We want to see the actual structure of the data where there might be two cell types. And for each cell type, we saw those cells in both replicate. This is what we want. But if we don't do anything to the data, the left-hand side will be what you get. So we need to do something about it. So there are several ideas that people propose to solve this batch effect problem. Let's start with the first idea. So if we assume that the batch effect that we are seeing is based on the low read count. So you can imagine that having a low read count is like a random sampling of which RNA molecules to sequence. If we have a million read count, then we essentially sequence almost everything. But if we have 20,000 read count, then we are basically sampling which RNA molecules that we are going to see. So that can be very random. That can be very highly variable. But because if we analyze the same cells, like the same cell type, when you see, let's say cell type A is a cell type. If you see cell type A in sample 1 or patient number 1, and you also see cell type A in patient number 2 or sample 2, because they are the same cell type, so the biology should be the same. But because of the sequencing process, introduce some noises. So transcriptomic profile that we get in sample 1 should be something like transcriptomic profile in sample 2 plus some noise from our sequencing run. But if you ask, like, if you pick cell type A in sample 1, fix it, and you go over all the cells in sample 2, got all the cells in sample 2, and you ask which cells in sample 2 is the most similar to my cell type A in sample 1. So you ask like which cells, maybe A or not A, like in sample 2, which one has the most transcriptomic with data compared to my cell type A in sample 1. Chances are your answer is still going to be cell of the same type. Because if you pick a different cell type, the transcriptomic profile should change so much that it no longer looks similar to A. But if you pick cell type A in sample 2, even if you add some noise due to the single cell analysis, they're still going to be similar to each other, somewhat. Not perfectly similar, but somewhat similar. So So people use this idea and develop a technique called mutual nearest neighbor. So this technique, so we have two samples from two batches, batch 1 in the bottom and batch 2 on top. In these two batches, some cells are from the same type, if we analyze the same tissue. So some cells we have the same type, the red cells here are the same type, the blue cells here are the same type, but purple cells might be unique to patient 1, and yellow cells might be unique to patient number 2. If we calculate the similarity in gene expression profile across the sample, across the batches, we're going to get some mapping. So each cell in sample 1 is going to point to some cells in sample 2. Each cell in sample 2 is going to point to some cells in sample 1. We get some mapping. What we are looking for is this kind of A point to B and B point to A. This kind of reciprocal mapping. So you have the arrow pointing from red to red, from red cell in batch 2 to red cell in batch 1, and also from red cell in batch 1 to red cell in batch 2. So this means that they are mutually similar. So compared to other cell types, the red cells are more similar to each other, even across batches. But if you are looking at cells that are unique in a certain batch, purple cells for example, it might look like red cells in batch 2 because that's the most similar. There is no purple cells in batch 2. It's going to point to red cells. But the red cell is not going to point back because red cells in batch 2 is going to be similar to red cells in batch 1. So you're going to get this distinction. If you have the same cell type, the arrows will point back and forth between each other. But if you have a cell from a different type, the arrow will only go one way. So people can analyze this pattern in the data and identify that, oh, okay, between batch 1 and batch 2, the red cells, this group, this cluster, the red clusters are similar to each other. They point back and forth between each other. And the blue cells are the same type. They point back and forth between each other. And the purple and the yellow doesn't belong to the other groups. So people call this a mutual nearest neighbor because nearest neighbors is represented by this arrow. Who is my nearest neighbor in the other batch? Red is the nearest neighbor of the red cells in the other batch. Okay. So this is similar. You might see this concept when you perform blast analysis. When you want to identify genes that are the same, like the same gene or the same protein, across organisms. What we want from blast result is that if we pick gene A from human, it should be more similar to gene A in mouse. And if we pick gene A in mouse, it should be more similar to gene A in human. This is also called reciprocal base hit or mutual nearest neighbor. So this can tell us that these two genes or these two cells that have the similarity mapping to back and forth between each other, these are the same cell type or the same gene. So that's one technique, one idea that we can use to map genes across batches. The other way, so the other two, I'm going to introduce 3 techniques here. The first one is easier to understand, so you know what to expect. The other two, they have some different ideas, so I want to introduce, but you don't have to worry if it sounds too complex. But I want you to try to imagine the algorithm as it applied to the data. Okay. So the second technique operate on a similar assumption that even if there is a batch effect, but the cell from the same type should still be similar to each other. So if you visualize the data, even though cells from different samples don't overlap with each other, but cells from the same type, by the symbol here, should still be similar to each other. So it should be nearby each other when you visualize the data. So cells from the same type should be near each other basically. So the second approach utilizes this assumption to say that if you do clustering So clustering is the process of grouping. If you try to cluster cells from multiple batches together, because cells from the same type, even though they are not on top of each other, they are next to each other. When you cluster, they should belong to the same cluster anyway, because they are next to each other. So once you identify the cluster, let's say you identify 4 clusters. In each cluster, let's say cluster 1 contains this cell type, which is present in, let's say, sample 1 and sample 2, but not sample 3. So inside cluster 1, you see cells from sample 1 and cells from sample 2. If you calculate the center or the centroid of cells from each sample, the green is one sample, purple is another sample. So if you calculate the centroid for the cells from the green sample and the centroid for the cells from the purple sample, they are not going to overlap, right? Because you still have the batch effect. But if the assumption is these are the same cell type, and in the end, once we integrate the data, we want them to overlap. So what we want to do to the data is we want to collect data from green sample, move it this way according to the green arrow, and data from the purple sample, move it this way, so that they are overlapped in the center of the cluster. And these techniques will modify gene expression, like scale it or add something to it, in order to move the data, move the gene expression profile from this location toward this direction for the green sample, and from this position toward this direction for the purple sample. So after one step, you are moving, the algorithm is moving gene expression profile from the green sample and the purple sample close to each other. So this might not work well, this might not finish in one step. So we move the cells a little bit. They might not completely overlap yet. So this process will repeat once we modify the gene expression data so that the cells become more overlapped. We can do the clustering again. Identify the cluster, identify the centroid for cells from different samples, and identify the direction to move them in order to bring them even closer together. And this process will repeat until everything converges. So at the end, you're going to get the number of clusters matching the number of cell types. And in each cluster, cell types which have the same cell type, cells from different batches, cells from different samples, are going to lay on top of each other. So this process will determine what kind of perturbation we need to perform on the green sample, on the yellow sample, on the purple sample, in order to make that data overlap. And this will then be applied to the original data, and you will get a new normalized gene expression profile for the three samples that are compatible with each other. So this is the second technique. So they do clustering and then update by assuming that cells in the same clusters are the same type and they should overlap. So we move the data bits by bits until the cells lay on top of each other. Okay. The third technique, this is even more convoluted. So there is a technique called canonical correlation analysis. Might not be familiar to many of you, but the idea is, so let's imagine, let's say you have a cohort of patients. When you study patients' data, you can do it from multiple aspects. You might ask for immune profile in the blood sample. You collect the blood sample from the patient. You analyze the immune profile. You can also measure the physical fitness of the patient, like physical properties of the patient. So these are two different descriptions. Like one is a molecular description, one is a physical description. But both of them describe the same patient cohort. So you have two measurement systems, like totally unrelated. But both of them are being used to describe the same samples. the same patient. Canonical correlation analysis is the technique where we want to understand the relationship between these two measurement systems. For example, like if one of them is an immune profile and the other one is a physical fitness of the patient. CCA, canonical correlation analysis, its goal is to identify if there is any link between certain immune parameters and certain physical parameters to identify the link between the parameters that are being measured. So maybe like T cell abundance might be related to muscle strength, for example. So this analysis tries to identify any link between the parameters. Okay. So to apply this idea to single cell data, so when we think about batch effect, we assume that we have the same sample, cells from the same tissue, but we analyze them on two batches. We can think of the two batches as two different observations, two different systems of describing the same tissue. So let's say we have liver cancer, we extract cancer liver tissue from the patient. Patient number 1, patient number 2. Both of them are liver tissue. So they should have the similar cell composition. We analyze them through single cell analysis twice, one at a time. So we have two different observations. And they are not the same because of the noise, because of the low read count. So in the context of CCA, we are thinking that, oh, these two samples have the same underlying cell composition. But we are describing it with different single cell run. Sample 1 or batch 1 can be thought of as one measurement system, and the batch 2 can be thought of as another system. So in this case, we want to identify correlation between the two batches that explain the same cell type composition. And if we can do this, that means we have a way of mapping data from one batch to data from another batch. Because CCA essentially is mapping one measurement system to another measurement to link the two together. In this case, we think of the measurement as batches. We think of the batches as a measurement. So we want to correlate them together, we want to link them together. So just to show like the diagram of how this idea is utilized. So let's say you have the same sample, same cell type. You treat them with drugs or untreated. Run single cell RNA sequencing, get two samples, treat and untreat. When you visualize the data without doing anything, they are going to look separated. But the same cell type will still be next to each other. So we can tell a little bit that there are probably 3 or 4 cell types. And we can tell that these cells from the treated sample and these cells from the untreated sample are probably the same type. But they are not overlapping. There's still some batches. So with CCA, they're going to analyze how the data from the treated and untreated are correlated to each other. So they're going to identify these kind of patterns. Doesn't have to be exactly this pattern, but it's going to show some mapping between cells from the untreated to cells on the treated. And you see a V structure, so you see that these cells from the untreated are probably the same cell type as these cells from the treated condition. And on the other side, you see some parallel structure between treated cells and untreated cells. So these are probably the same cell type. But the limitation of CCA, this is correlation. CCA is correlation analysis. So when you think about correlation doesn't mean equal. You can have two things that are correlated, but they have different magnitude. So this V-shape can also summarize the fact that after correlation analysis, you see this association between cells from the two conditions. But they're still not on top of each other. But they are now aligned with each other. They are correlated with each other, but not yet on top of each other. So you need another technique. This is from electrical engineering. If you have two signals that are parallel, but not exactly equal, you can perturbed, you can adjust one signal to match the other. And this dynamic time warping, you can understand it from the name. So they're going to assume that this V shape kind of map the cell type from one cell type to another cell type. If you think of this as a signal over time, one cell type being 0, and as time goes higher and higher, the signal changes into a different cell type. So you have two time signals, one for the untreated condition and one for the treated condition. And based on the variance, you can see that the untreated, the blue cells have more variance, so it takes longer time for you to go from one cell type to another cell type. For the treated, it's more clustered, so this requires less time for you to go from one cell type to another cell type. So dynamic time warping try to kind of unravel the treated cells here to make them as scattered as the untreated cells, to match them along this path. And at the end, when you combine these two techniques together, you use CCA to understand the correlation structure between the two batches, and use dynamic time warping to make them not only correlated but equal. At the end, you're going to get something similar to the other two techniques, where you move data from one batch, you perturb the data from one batch to make it match the data from the other batch. And at the end, no matter how many samples you have, what we want after integration, we want to see data being on top of each other if they are from the same tissue. We don't want to see them separate. Okay. And to expand further, this idea that we can integrate data from two batches together. So the technique that we have mentioned so far, especially the canonical correlation techniques, they don't even assume the two measurements to be the same omics. Right now we are talking about mostly transcriptomic RNA sequencing. So we want to merge RNA sequencing from batch 1 to RNA sequencing from batch 2. But these techniques, they are not restricting the two batches to be the same omics type. So what people did, very clever with this idea, is that if you do multi-omics at single cell level, so let's say you perform RNA sequencing, and you also perform ATAC-seq to understand the open or close, the accessibility of the chromatin. You can identify cell type from RNA sequencing data because you know gene cell type markers at the transcript level. You know that B cells express certain genes. But when you perform ATAC sequencing or any other omics, there's no signature to know if B cells have accessible chromatin somewhere, or B cells have methylation profile somewhere. We don't have this kind of knowledge to map cell type to omics other than transcriptomics or proteomics. So when you get the data, it will look like this. For the RNA-seq, you can map all the immune cell type. But for ATAC-seq, you cannot do anything. They are just cells. But because we have techniques that can integrate across batches, in this case, because chromatin accessibility, whether it's open or closed, is going to be somewhat correlated with gene expression. If the chromatin is open, you can have active transcription. If the chromatin is closed, you probably cannot transcribe the gene. So in a sense, chromatin accessibility can be thought of as a measurement of gene expression, a noisy measurement of gene expression. So we can think of this as a new batch of the data describing gene expression in a noisy way. So people use the same idea to integrate ATAC-seq together with RNA-seq. And by doing that, we can map all the cell type that we know from RNA sequencing data onto ATAC sequencing data. So this allows us to do multi-omics on single cell. Very easy. Okay. So this is it for now for single cell. Next session we're going to talk more. So let me introduce you to other techniques that allow us to analyze tissues or cell culture at high resolution. So in some situation, so single cell it requires you to be able to isolate all the cells. But in some tissue, depending on the structure, depending on the cell attachment, you cannot isolate them easily. For example, in fat cells or in neuron cells, you cannot easily isolate individual neurons or individual adipocytes from the tissue. So in this case, what you have to do instead is to do a single nucleus sequencing. So as the name implies, so when you single cell, you are isolating the cells and you get the RNA in the cytoplasm. But because for certain tissue structure, you cannot isolate individual cells, but you can still isolate the nucleus because of the you can still isolate the nucleus. So this single nucleus sequencing allows you to study RNA pattern and gene expression pattern in cells in tissue where you cannot isolate. But the limitation is you are going to see only RNA that are present in the nucleus. And we know that RNA are exported into cytoplasm for translation. So the amount of RNA in the nucleus is not going to be very high. But again, this is good enough if you want to distinguish the cell type. So people do this as a last resort for tissue type that where single cell techniques are not applicable. Another cool application of single cell technique is if you want to study evolution of the cells, if you introduce some maybe radiation or maybe any compound that induce mutation, and you want to see how the cells adapt, you want to monitor the mutation. If you want to monitor the mutation, if you do exome sequencing, that's a big investment. So people came up with a way to do lineage tracing. We want to know which single cells are derived or are daughters from which single cell. We want to know their lineage, how they evolve, how they change. Do the daughter cells gain a new phenotype, or is the expression level in the daughter cell change from the ancestor? We want to know the change. So we need to have a way to track which cells derive from which cells. So in this case, they developed a cassette of DNA called cell tag. So this cell tag is going to have an expressible, so it's like a gene with its own sequence. But it also contains a unique nucleotide tag that will be unique for each cell, because now we are tagging individual cells. So individual cells will get this expression cassette. So it will express this gene. But the gene sequence that each cell gets will be different. So when we run the RNA sequencing, we can detect the expression of this gene. And we can look at the reads, look at the sequence to identify the evolution pattern, because these nucleotides, as the cells divide, mutation will accumulate. So you can map which cells are related by looking at the similarity in this region. So you can trace how the cells change, how the expression change over the generation in response to your treatment. So this is one of the cool techniques that once you have the ability to monitor single cells, you can even trace how they divide and their phenotype over time. Okay, the last part. So we already talked about single cells. So single cell is good in the sense that it gives you high resolution of your tissue structure. But it doesn't actually give you the structure. Single cells give you the composition. Because once you isolate all the single cells from the tissues, you no longer know where they are in your original structure. And some people, in some disease, in some situation, we want to understand not only the composition of the cells, we want to know how they form tissue. Right? Because even when we know the composition, who knows, maybe in the tissues, all the same type, all the same cell type are grouped together, or maybe they are all scattered and form many heterogeneous areas. So people want to understand not only expression data for individual cells, they want to understand where they come from in the tissue, in the organ. So the most straightforward way would be to, so with single cells, like we barcode all the individual cells. So we can also add another barcode to the location on the tissue. So we can have like a micro needle to sample cells from different locations in the tissues, barcode them with different tags, and then analyze the data. Now we not only have the cell tags, we also have the location tags. So we can reconstruct the structure of the tissue by looking at the location tag. But you can imagine that this will require a lot of work. Basically, you're going to sample, if you want to study 1,000 sites on the tissues, you're going to sample 1,000 times on the tissue. So people try to come up with other ways that they can analyze gene expression or protein expression on the tissue without having to actually sample the tissue. Can we do it on the intact tissue itself? So now this becomes an imaging method. So we don't want to sample the data and run sequencing, but we still want to see gene expression on the tissue. So people do this with imaging, and this is really cool. So you probably all know about fluorescence kind of staining. So we can stain certain gene, certain RNA, or certain protein. We can activate the fluorescence, and we can take the image, and now we can see the expression of certain genes or proteins on the tissue. The limitation in the past was that you don't have so many fluorescence dye. You cannot tag so many proteins without starting to have the same color mapping to multiple proteins, multiple genes, or you have interference among fluorescent signals. So people want to develop a better way to tag the gene, tag the protein in the tissue. And you might be surprised that nowadays we can visualize more than 10,000 species of RNA in a single tissue. So basically you are getting close to having RNA sequencing level data onto the tissue with just imaging, no sequencing involved. So the method that I pick here are representative of many ways that people try to solve this problem. How to visualize a large number of isoforms, genes, proteins on the tissue. So this technique that enables like 10,000 RNA species to be visualized, the idea is very cool. So okay, let me use this diagram. So basically, they're going to use a hybridization method. So this is nothing new, right? Hybridization. You have a probe with a fluorescent. When it hybridizes to the RNA, then you get the signal that tells you where the RNA is. So this is similar to nanostring, right? We talked about nanostring before. But nanostring, what they try to do is they create a barcode of fluorescent signal. Green, red, green, blue, blue. Each one being linked to one gene. So that you can visualize different genes at the same time. But the limitation of nanostring is that you can only have so much combination of fluorescent onto the same probe. So people thought, why don't we create a combination of fluorescence but across multiple probes? So the first idea that the first step is we have a gene. A gene is really quite long. In nanostring, you are targeting with only one probe to target that gene. But who's to prevent you to design multiple probes per gene, like microarray? In microarray, we design multiple probes per gene to get more hybridization. In this case, they're going to design their probes so that each gene can be bound to multiple probes. So at the end, so if we focus on the diagram here, so at the end, each gene, each RNA molecules will be bound to multiple probes. But if you let all the probes bind at once, you are not gonna get many different signals, right? Because you're gonna get green yellow yellow, right? Green yellow red. When they are close to each other, you might not be able to distinguish green yellow red from red green yellow. They are probably gonna look the same as a combination of three colors. So in this case, that technique is called sequential hybridization. So this is similar to if we think about sequencing process inside Illumina. Right? If we want to know the sequence for a DNA molecule, we incorporate nucleotide one at a time, and we look at the color. If the color changes from red to green to blue, we know the DNA sequence. But in this case, these people design their fluorescent probe in such a way that in the first round of hybridization, this probe will bind to certain genes and they will have certain colors. So in the first round, we already have this signal. This gene has red, this gene has yellow, this gene has yellow. These can be different genes or the same gene, we don't know yet. But then they introduce a new batch of probes. And these batches are designed to bind the same set of genes, but they have different colors. So the first gene will be bound to red probe first, and then green second. Some probe will be bound to yellow first, and then green second. Some probe will be bound to yellow first, and then blue second. So as they repeat this set, each round of hybridization, they introduce new set of probes that are designed to target specific gene and have specific colors. So at the end, they can decode the color in the same way that Illumina decode that DNA sequence by looking at the sequence of colors from different hybridization rounds. And they can say that if we see green, blue, red, yellow, then this is gene number one. So the possibility is now endless. As you increase more rounds of hybridization and you design more probes that can specifically hybridize to specific genes with specific colors, the number of possible genes you can probe is exponential. So basically the number of fluorescent to the power of number of hybridization rounds. So with this technique, you can follow more than 10,000 RNA species, and each of them will have a unique fluorescent signal over these hybridization rounds. And this now the limitation became about the imaging. Now we can produce very detailed fluorescent signal for individual RNA molecules. So the next would be they have to combine this with a very powerful microscope so that they can isolate individual dots inside the cell. So this is a cell, and each dot is the RNA that they can tag. So you can see how dense the information is. So you can think of nanosync idea combined with Illumina sequential signal, combine the two together, and you get a way to visualize all the RNA inside the molecule. For protein, we use immunofluorescence antibody. So people also have to come up with ways to repeatedly label different proteins, similar to different round hybridization. They have to come up with ways to repeatedly monitor different proteins. And they do this with a cyclic reaction. So they attach antibody to the protein, and then they clip it with enzyme. So they remove the antibody. And then they attach it again, but to different protein, and then clip it. So with this, you can target like hundreds of proteins in the single tissue, but you have to do it in cycle. So the only thing that this pipeline needs is time and money to repeat the reaction process. And just to show you that what we have today, we are moving toward 3D information. So we know that the tissue is not 2D, right? It's 3D. So nowadays, people are analyzing using imaging combined with hybridization and antibody to visualize the expression of gene and protein, and then put all the data together to see the 3D structure of the tissue. So this is like the new future for. So we go from bulk omics, where we only see like the average. We collapse all the cells in the tissue into just one set of numbers, to single cell, where we isolate all the individual cells and we analyze them separately, so we know the composition. And to spatial techniques, where we now start to probe gene expression or protein expression on the tissue structure itself, either in 2D or 3D, so that we now know whether some cells are next to each other or are they scattered inside the tissue. So these are really important to understand like the structure of the organ or the structure of the disease inside the tissue. Okay. So just to summarize, single cells and spatial techniques help us understand the heterogeneity of the tissues. Single cell data provide composition, but no location information. So currently people are trying to integrate the two. So single cell sequencing has sensitivity because it do sequencing for individual cells, but it lacks the structure information. Spatial techniques, imaging techniques, have the structure information, but with imaging, you cannot see the majority of the RNA molecules. So you still need single cell techniques to get the measurement, to get the read count, to get the molecule counts of the genes. So people are seeing that integrating both of these aspects will be the future of omics. Okay, so that's everything I want to talk about today.